Good afternoon, everybody. Chairpersons, colleagues and friends. At the outset, I would like to express my sincere thanks to Dr. Rajiv Arshni for inviting me to be here this afternoon to present our work. I have chosen a very specific topic instead of presenting whatever we are doing in the lab and covering a broader spectrum of activities. Last time, I was also fortunate to be here to learn from the international gathering, experts, so much of development that is taking place. I learned from that, and so also this time. And last year, I presented our work on the mutants that we have been doing. We did, over a period of 6 to 7 years. So this specific topic which I have chosen this time, to say very briefly, what is that we could do using genome-wide approach to address salinity tolerance in rice. India worries much more than anybody else because this is very frightening. It's going to be, as it is predicted, that going to be the most populous country in the world. By 2050, it would be having about 1.6 billion population, more than that of China. So that demands that we have to address the issues far more urgently than anybody else in the world. And this is further complicated. This is being talked about for past 2 days. The climate is changing, and India would be in a far more disadvantageous position than anybody else with regard to the impact, negative impact that is going to encounter in years to come because of the climate change that is happening. And so we all know that we have been talking that abiotic stresses do impact, and as a part of climate change and otherwise this is happening. Drought, excess water, particularly in case of rice, salinity, sodicity, acidity, heavy metal toxicity, and so on and so forth. And particularly in eastern India, where rice cultivation predominates, there is a problem of a low light intensity problem as a biotic stress. So these stresses often combine. For instance, the submergence tolerance and drought, submergence and drought, they occur in the same season. The crop encounters both the problems. And similarly, heavy metal toxicity and mineral deficiencies go together. So the salinity is an issue which I am going to describe. So this work has been carried out, a group of people, and these names are mentioned in the abstract, although in this presentation you see my name alone, but at the end I will acknowledge, but in the abstract all the names are there, and only one co-author is present here, Dr. Swarup Parida, who is present here somewhere, so who contributed the major portion of the work which I am presenting. So the salinity is the major problem worldwide, and 12 million hectares, this is estimated area, and 50% of that in India itself. And salinity certainly affects very significantly, starting with germination, seedling growth, and then also major yield components getting influenced negatively, particularly the spikelate number. And that impacts the yield, ultimate yield, and loss ranging from 27% to 50%. So that's what has been assessed, particularly when it occurs at reproductive stage. And worldwide, if you analyze, most of the publications on QTL mapping, there are not many papers on association mapping for salinity tolerance in rice, but QTL mapping papers, they address particularly the vegetative stage salinity stress. So reproductive stage salinity is far more challenging because of the phenotyping issues, and we all know that quantitative in nature, salinity, stress, tolerance, many loci, and several QTLs are known, and of course there are unequal contributions and influence of different loci, varying degree of epistatic interactions that take place, and also QTL environment interaction often strong. Conventional breeding has been dealing with this problem for a long time. It's not that, with genomic revolution, we want to have a altogether different ways and means to address this problem. Maybe you would be able to supplement this in a great way. There have been pure line, pedigree, and backcross breeding efforts in the country and elsewhere to develop varieties, which are salt tolerant. Biparental, multiparental populations have been developed, and also phenotypic selection largely has been carried out in the country and elsewhere. And there are varieties in the country and also elsewhere which have been developed. For instance, CSR30, CSR36, and so on and so forth. So to summarize that the conventional breeding efforts have been successful to develop varieties and which are working in the field, but they do not have a very high level of tolerance, particularly the dry season tolerance. And coastal ecosystem, you have wet season salinity and dry season salinity, and one finds in dry season one requires much higher level of tolerance than wet season. And some of the areas where very high level of salinity occurs, these developed varieties do not really work. But the land races do work, but they yield very poorly. So that's the reason why there is a need to really work on using all the tools and techniques which are available at our disposal to address the problems. And the IRI has been working for quite some time on this particular issue. And this is one area, Michael is here, and he did work on this, and then several QTLs identified, and this is one locus which has been targeted. The markers have been identified, and 3 loci, in fact, genes have been cloned in this particular region, and which is on chromosome 1, and this is called salt region on chromosome 1. And one can see, for instance, SKC1, which was cloned some years before, almost 5 years ago, and there is also SALT-T, which was even cloned much before, and also there is another called pectin esterase, located somewhere here. And so these are 3 genes, and gene-based markers has been developed at IRI, and some of these markers are being utilized by national programs. And this line, for in particular the FL478, which has been developed through backcross breeding, the loci has been transferred. This particular region have been introgressed from Pokali, the salt-tolerant types which are available and in India, and it has gone to this background of IR29, and then this line is being utilized as a donor in backcross breeding programs. And there are advanced breeding lines which have been developed in the country and elsewhere, and particularly transferring this particular segment which is highlighted here, and at least these 2 to 3 genes which have been integrated and introgressed to popular background. So while this is going on, this provides an opportunity, because you have one particular region which is controlling a large part of phenotypic variance, but also at the same time challenges to characterize this further and verify, particularly because all these are under characterized particularly at the vegetative stage, and what happens at the reproductive stage. Some early studies suggest that there is a dichotomy between the tolerance that we encounter to see at vegetative stage and that we see at reproductive stage. And we are approaching this particular problem, trying to address this particular problem from different angles. One, we have biparental populations, and then we are trying to map the QTLs as it has been done by many workers. In fact, more than 60 to 70 quantitative trait loci have been identified, mapped on different chromosomes of rice for salt tolerance, and most of them with regard to vegetative stage tolerance. That is one area we are working, and I will not actually present much of data on that. Second, we have a mutant population. Professor R.P. Sharma is here, who coordinates this activity, more than 20,000 lines, and we have screened these lines, a part of it, of course, and identified some of these mutants, and which have varying degree of tolerance to salinity. And we are characterizing those to understand how it operates. Also, we are carrying out the transcriptome profiling and sequencing, and also microarray-based profiling. Not only salt tolerance, also other abiotic stress problems. For instance, in this case, moisture deficit stress, Nagina 22 and Dagardesi. There are land races from India, and submergence FR13A, salinity, Pokali, CSR 30 and Nona Bokra, and heat, Nagina 22 internationally known, and Konarulu. So we are taking this and taking contrasting susceptible types. We are trying to understand gene expression pattern and to work out the common pathways which are operating across stress scenarios, so that if you manipulate some of those common genes, you will be able to manipulate multiple stress tolerance. So Professor J.P. Khurana from Delhi University is working actively and carrying out this particular, most of this work. And we did also part of this work, particularly with regard to salt tolerance. That's what I'm going to present briefly. That's what we chose, because they are phenotypically very, very close. And it is supposed to be a backcross derivative of CSR 30 is supposed to be a backcross derivative of Taraudi Basmati. So both are having Basmati quality traits, and CSR 30 is tolerant and Taraudi Basmati is susceptible. And so we chose this carefully because the genetic background effect would not be much disturbing, because when you do take unrelated genotypes, one gets thousands of genes upregulated and downregulated. One gets lost in the process in choosing the right kind of candidates for further analysis. So by choosing these genotypes which are very close in terms of genetic background, we could identify, and we imposed trace and did the transcriptome sequencing and identified genes and several millions of paired Sulexa transcript reads and mapping those, and then doing analysis further. And that's what we identified in case of the control versus trace. In case of the tolerant one, 1,274 of them were identified, and then in case of susceptible one, also almost equivalent number, 1130 upregulated, downregulated, differentially expressing ones were identified. And one gets, there are, out of these 986 are common, and the tolerance specific, 288 of them, which are very specifically expressing in CSR 30. There are also some specific ones expressing in the susceptible one. And further other characterization also we did subsequent to this. And you can see some of the categories of these genes, functional annotation of about 288 of those which are differentially expressing in the tolerant one. So that's what, specifically expressing, differentially expressing, and that's what you see. And you can see all the important genes, gene classes are noted down here. And for every class you find few genes showing differential expression, and 54 of them. And these are the 54 known clone genes which are already known to be operating in this. And there are also 192 candidate genes which are known to be specifically expressed. And then there are also additionally unknown expressed genes. And if one take this together, about 85% of those known salinity stress responsive genes, and for which 128 of them very well the pathways are known. For instance, SOS pathway, 79 of them signaling pathways, 32 of them, and so on and so forth. So what does it really indicate? It indicates that when stress is imposed, so all kinds of genes are getting differentially expressed and cutting across pathways and the gene classes. And that's what indicated here. There are classified further particularly 246 of them, and then which are specifically differentially expressed in the tolerant one, and I'll not really go into those details. Transcription factors and signaling components and kinases and membrane proteins and so on and so forth. So many genes getting differentially. That too, there are close relatives, genetically very close. So that's what one sees. So we also, when we compared our sequence information, we did sequencing of these two. I'll present this data after the slide. And we did find single nucleotide polymorphisms and insertion deletions, and which are in these many genes, which differential expressing genes, and which we thought of utilizing subsequently in our genetic association studies, genome-wide association studies. And there is ABA-dependent pathway, ABA-independent pathway with regard to osmotic stress, and then there are other pathways, ROS pathway, and then calcium PLC pathway, SOS pathway, and all these pathways, and so many genes, and many of those important genes, and some of those we discovered, and many of you also keep describing this, and that provides a so complex scenario to deal with this. Are we to modify and deal with each one of them? Or we have to only pick few of them and which one to pick and then go ahead in breeding programs? Or we should go for genomic selection targeting all these hundreds of those genes and then ultimately define what is that gain we are going to get through a training population? What is that approach we have to really follow? That provides a very complex scenario, and I believe there cannot be a better gathering to provide a strategy to deal with a complex traits like this, where hundreds of different genes and all appear to be important to be operating. And when we put our genes into different categories, for instance, and we do find that, for instance, SKC type, SKT1, for instance, that particular class, there are 7 of them, and and 40 of them potassium-sodium homeostasis system, and SOS1, 5 of them. So they're already described, and you can see cytosolic ones. There are several oxidative stress, and then different other categories, osmoprotectants. You have those genes which are mentioned here. So all these have been shown to be differentially expressing. And so also the nucleus for the DNA binding, and then transcription factors, and chloroplastic, mitochondrial, you name a thing, and then so all of them appear to be showing differential expression, and one expects that they will be certainly involved in this. And similarly, 44 differential express genes in salt to overly sensitive pathway, and then calcium signaling pathway. For instance, 19 of them here, and then almost 25 of them here, the tonoplastic and elsewhere. So all of them are showing to be differentially expressed and also involved in this. So mapping based on the SNPs which are there in these genes, and try to understand how these genes can be implicated, at least short listed for the purpose of breeding. We did resequencing of some of these contrasting types, and then which differ. For instance, CSR30 and Tharadi Basmati, and then this is typical Basmati which have been sequenced, and then we are yet to publish this. But then when we compare this, certainly we found many single nucleotide polymorphism existing. Mind you, they are phenotypically very close. They look alike almost, very difficult to differentiate. And the Basmati quality characters are almost identical between these two. But we do find, SNPs, several thousand SNPs, rather lakhs, and then this is the frequency, 1.9 SNP per kb, and there are almost 12,000, more than 12,000 genes carrying 37,000 single nucleotide polymorphisms, non-synonymous and regulatory SNPs, and so on and so forth. So large number of SNPs which are there differentiating these two so-called very similar types. That was a little surprising. And we designed Infinium assay using this, and we chose 6,000 of these. Majority of them responsive under abiotic stress, and also some for abiotic stress, and some of them to fill the gap. And this is a kind of classification: abiotic stress responsive, biotic stress responsive, and other categories. And this is a kind of distribution, a genome-wide distribution of these 6,000 chromosome wise, which is given here, and also you can see the distribution. Minimum is about 15 kilobase and maximum is about 98 kilobase, and average is about 51 kilobase. That's the kind of distance between two adjacent single nucleotide polymorphism markers as they are distributed across the chromosomes over the genome. And by taking this, we did. And you can see initially we used this, a subset of this, to understand differentiation of upland types from the lowland irrigated ecosystem rice varieties, and this we published in BMC Genomics 2-3 years ago. And you can see the upland and lowland ecology types are differentiated by very clearly, for instance, 9 SNPs and 4 in missense, non-synonymous substitution, and 5 of them creating premature termination codon. So this encouraged us to analyze it further for salt tolerance. And so this you can see, Wernicke transcription factor at the DNA binding domain. There is a single nucleotide substitution A to C, and that differentiates the upland types from the lowland types. And very important, finding, and we are further validating this in larger sets of germplasm lines. And this we published in BMC Genomics, and we extended this, taking the 6K chip, and recently this is coming in DNA research, and we did a phenotyping of 220 rice accessions in detail for their response under salt stress. These are the different conditions and way it was phenotyped. I'm not really going to describe each one of them. And with this, and time is running out, this is the kind of distribution of these 14 different traits that we handle. And then this is the kind of genotyping results on LD that we see here. And very briefly, the overall LD for SNP at 20 kb distance from each other was about 0.5, and which decayed to half value at about 300 kb. And more importantly, the regions of LD with the significant p-values were found to be interspersed, in the region, the whole genome. And in different groups, for instance, this is the indica non-aromatic type, and they differed, although the Aus type and then aromatic type similar pattern, but the indica group differed very significantly from the other groups. And this is through the association, genome-wide association. The details can be seen in the paper, which is already online. I'll be very brief. So what we found is there are loci, and I'll go to the next one. You can see across the different traits that we have chosen, and you find different SNPs which are showing very significant association, and this is the kind of percentage of phenotypic variance which are explained by the loci. And we knew when we corresponded these loci to the known QTLs which have been mapped, and more than 70 QTLs have been localized, and large number of them, particularly vegetative stage, salinity tolerance. And this is Michael's paper, and which was published in Rice some years before. And you can see some of these loci, one here, 1, 2, 3, 4, 5, 6, 7 of them are presented here. And our loci which are identified through association corresponds. For instance, this one largely corresponds. This is the Saltall locus on chromosome 1, and similarly on chromosome 4, and similarly on chromosome 6. So these these three, corresponding to the one which have been reported earlier. And similarly, there are several other papers, and we did find correspondence with the QTLs which have been reported earlier based on biparental mapping populations, and that provided confidence to us that probably whatever mapping we did, it does work, although only 220 individuals were used and a 6K chip was used. So it tells us that 6K chip, based on the stress responsive genes, worked very well and identified some of these known QTL locations. But in addition to this, also we identified new regions because these 220 lines represent a diverse set representing different mechanisms of salt tolerance. And that's the reason why we identified also the new ones. So I'm not really going to those details of new types. And this is the kind of salt tolerant region. And more than 12 single nucleotide polymorphism showed very strong association in this region. And we know also that some of these loci which we find also correspond to candidate gene regions. And for instance, on chromosome 1, as I said, there are the two known genes which are cloned already in addition to SOL-T. And similarly, on chromosome 1, there was another which was showing very strong association. It corresponded to NAC6 and NAC2, which are already implicated in drought, salt, and cold tolerance. And similarly, another one on chromosome 1, salt stress root-specific protein, which have been already reported, and one on chromosome 9, which is corresponding to delayed seed germination. So candidate genes which are either carrying the SNP or very close to the SNP probably can be further targeted, to pyramid them. And the recent study also says that these genes through associations have been confirmed to be showing very strong association with salt tolerance. For instance, SOL-T, which was cloned years before, and is concerning expression and production of osmoprotectants. And this is located in the salt-tolerant region on chromosome 1. And similarly, SKC1 region, and concerning maintenance of low sodium-potassium ratio, because it removes sodium, throws out of the xylem, and that's how maintains higher potassium ratio. And similarly, others. So they are known, they have been already confirmed and demonstrated through association analysis that they are relevant. And more so, this paper says that single nucleotide polymorphism, which is present in this set of genes, are relevant. And most likely, these SNPs are involved in differential interaction. But that part is very poorly understood with regard to salt tolerance. We don't really understand so much the interactome, how these genes interact. And that would be a very interesting story to really unravel that what is happening at the interaction level among these set of loci, which are shortlisted and showing very strong association. But the important point is that 220 lines which we have taken, although it represents a diverse set, but if we change that, certainly we will be dealing with some other additional loci. So there's a need to really expand this set of lines, and so that one identifies additional regions. But more than that, it is not really enough to say that those which are present in coding regions of these stress-responsive genes, they will be responsible and must be targeted. But more than that, differential expression tells there has to be differences in the regulatory regions of these genes. In fact, we did some analysis with regard to abiotic stress-responsive genes, and then we did find out that this single nucleotide polymorphism, for instance, if you take this unknown express gene and analyze this regulatory region, one finds, for instance, ethylene response element in this particular case. This is particularly drought tolerance, not salt tolerance. I am showing this for the sake of example, and one finds in this particular ERE element there is a single nucleotide polymorphism. Nagina 22 is drought tolerant. IR64 is irrigated ecosystem variety, drought susceptible, and there is a T2G difference between these two. And vis-a-vis this, if you analyze the expression pattern, what one finds is this is IR64 control, this is IR64 stress. Unfortunately, this is not coming. So IR64 control this gene gets down-regulated if you impose stress on this. But in contrast to that, if you take Nagina 22, this gene normally is very poorly expressed, but if you impose stress, this gets upregulated. And that happens for a large number of genes which are showing differential expression. And, and the upstream regulatory regions, if you analyze, you do find there is a correspondence. And some of them are implicated in drought tolerance, these candidate genes. In contrast to this, and if you see those genes which carry SNP in their coding region, coding SNPs, obviously the differential expression doesn't really correspond to the SNP side, but certainly these SNPs, as it was said in the previous one, must be doing something downstream at the interaction level. So there is a need to address both the issues, that differential expression and SNP in the coding region, and how both can be integrated to address and select some of those to understand the trait further. So this is summary, and so SNP based on stress responsive genes did provide opportunity to identify newer ones, newer loci, and verify already identified regions which are known to influence salinity tolerance in rice. Genome-wide association study enabled to identify, using this 6K chip, to identify new loci concerning salt tolerance, taking a set of 220 diverse lines which were characterized for reproductive stage. This has to be underlined, reproductive stage performance, which they were exposed to salt tolerance later stage. And the correspondence was evident between vegetative and reproductive stage loci, which was earlier identified at vegetative stage. They were also confirmed, at least saltal locus. And saltal emerged as a major locus even for reproductive stage salinity tolerance. And possibly because there are several genes located there, and they have been intergressed, that region has been intergressed from focally and getting also transferred, and contains number of genes and possibly represent a co-adapted gene complex segment, and all the genes are co-inherited and going together and controlling expression of this particular trait. There could be other regions and together influencing this. And coding as well as a regulatory region SNPs as discussed are very relevant from the viewpoint of establishing association and very specifically choosing from these genes which are showing differential expression. And in view of many genes involved, it is worthwhile to understand the salinity tolerance interactome, and that would provide opportunity to understand the trait expression better in different genotypes and how it changes, the interaction changes in different genotypes, and then further sharpen our strategy, redefine our strategy, refine our strategy so that we can deal with this particular complex trait with a lesser number of genes, and so that breeders can work on this happily. And with this, they are the people who contributed. Dr. N.K. Singh, this work was carried out earlier at my earlier institute, Indian Agriculture Research Institute Biotechnology Center, and then Swarup Parida is sitting here, contributed largely to expression and other work. And Vinod is the first author of this DNA research paper, and other collaborators. And they are the people who did phenotyping at the Central Soil Salinity Research Institute, Karnal, Dr. Sharma and Krishnamurthy. And I thank you very much, all of you, for your patience hearing. And once again to chairpersons for giving me sufficient time to complete my presentation.
And
lastly to Rajiv once again for giving me this opportunity to come here and present very specifically on our work on genome-wide association analysis. Thank you very much once again.
Okay, thank you. I think Trilochan will be available afterwards for questions. I think we might need to move on. Thank you.
Thank you very much.
Okay.